already jumped ahead. All right, we're going right in. I'm going to be talking to you today about the Cancer Genome Atlas and Nexus version 7.0. A little background about the Cancer Genome Atlas. I'm sure most of you are already aware, but the mission of the Cancer Genome Atlas, or TCGA, is to understand the molecular basis of cancer through the application of genome analysis technology. All of the data that's generated is publicly available with the ongoing continuous addition of new samples. Currently, there are multiple different cancer types with data available, and this includes breast, brain, kidney, colon, prostate, and ovarian cancer. For all of these different cancer types, there's different sorts of data available, and this includes, but is not limited to, sequencing of both DNA and RNA, expression of mRNA and microRNA, copy number information, and methylation information. For the data, the results I'll be presenting to you today, I'm looking at the colon adenocarcinoma, or COAD project. Colon adenocarcinoma, also known as colorectal cancer, is the third most commonly diagnosed cancer among men and women and is attributable to about 9% of all cancer deaths. There are inherited gene mutations which have an increased risk of colon cancer, and this includes both FAP and HNPCC, or Lynch syndrome. And there are also a number of factors for the sporadic incidence of colon cancer, and this includes increasing age, obesity, a low-fiber diet, diabetes, smoking, and colon polyps. So for the TCGA COAD project, I analyze this data using Nexus. Here I'm showing you a simple workflow of the processing. So first we take the raw data and pre-process it within Nexus to get the log ratio value for the probes, and then segment that data using the algorithms within the software. From that, we identify regions with copy number change, genes within those regions, and then ultimately the functional relevance of those changes. Today, I'll be presenting to you the level 1 data. This is raw Affymetrix cell data. It is available upon request from the TCGA. They also have level 3 pre-segmented array data, and this is publicly available to anybody who wishes to get it, and this will be uploaded already in the segmentation step. I will also be presenting information with the level 2 MAF files. This is a somatic mutation file. And again, this is also publicly available to anybody who wants to download it. And these include all of the mutations, but only somatic mutations, no germline mutations. If you are interested in getting this raw or segmented data yourself, you can get it through the TCGA data portal. I have the URL listed right here at the bottom. And we also have within Nexus DB the pre-segmented SNP array data as well. So one of the reasons that I specifically am using this raw data is that it allows us to do pre-processing of the data within Nexus, and also allows us to use our in-house algorithm for the segmentation. And I'd just like to take you briefly through some of the pre-processing that we've done to this data set. The first is match paired analysis, and just to explain what that means, for every sample we have copy number changes for both the normal and the tumor. Here you can see blue for gains. In the small region right here, there's a red loss. That's in both the tumor and the normal. And then there's this larger copy number gain that's just in the tumor. When we do a paired analysis, we remove all the calls that are in we basically subtract the normal calls from the tumor, so that only these somatic tumor-only calls remain. So all of the samples that we'll be looking at today have undergone this pairwise analysis. All of the data has undergone systematic correction. This is also sometimes called GC wave correction. And what you can see is that you can sometimes have this aberrant waviness that occurs. This is due to the local GC content of the individual probes at the genomic level. And this can introduce a lot of false calling. So after applying GC wave correction, we see that the data is smoothed out, and we get we remove these false calls. GC wave correction does not introduce waviness. If your data is already flat, it simply removes any waviness that may be giving aberrant calling. And finally, all of these samples have undergone ploidy correction. And I'd just like to explain this in a little bit more detail. So if we look at a linear genome view of an example sample, you can see we have red for loss with the corresponding yellow for allelic imbalance. We have blue indicating regions of gain, and we have purple indicating allelic imbalance, which commonly co-occurs with the copy number gain. But what you'll notice is that for all of the areas with single copy gain, which are marked out with the arrows here, we do not observe the co-occurring allelic imbalance. If we look at the drill down to the probe level view, what you can see here, I'm showing you again the linear genome view as before on the top here. The second panel is the log R values for all of the probes, and then the bottom panel is the B allele frequency values. And so I'd like to draw your attention to the bottom panel first. When we are looking at the B allele frequency values, for a normal balanced heterozygous state, we typically expect to see a 3-band pattern, which we can see here for the AA, AB, and BB alleles. And you can see we see that on chromosome 2, 5, 7, 9, 10, etc. If we now draw our attention upward to the top panel, you can see that all of these balanced heterozygous states are actually corresponding to this, this yellow bar right here, which is right above the single copy gain threshold. So you can see here again we have chromosome 2, 5, 7, 9, and 10, and so on, are all showing single copy gain. And if you look further, you'll see that actually nothing is being called at this baseline state. So this is an example where the recentering is wrong and we need to make an adjustment. Within Nexus, we can manually call these regions, so by calling chromosomes 2, 5, 7, 9, 10, etc., and labeling those as diploid regions, we can adjust the values and recenter these samples. So what's happened here is this bottom panel has remained unchanged, but the top panel, everything has been moved down. So you can see that now these are at the baseline, these diploid regions, and if we look at the summary, you can see we still have red loss with yellow loss of heterozygosity, and we have now this area as a single copy gain with the purple allelic imbalance, and all of these areas where we saw the balanced heterozygous 3-band pattern are now being called as baseline 2 copies, which is exactly as we expect. So for all of these samples, we've done this correction. If you're interested in reading more about these preprocessing steps or other preprocessing methods, we have these posted on our blog. The URL is listed right there, blog.biodiscovery.com. Also, if you're interested in hearing more about the copy number calling algorithm, you can view our webinar archive, as well as any software information you might be interested in on our main webpage, biodiscovery.com. Okay, and now I'm going to switch over to a live demo of the software. Okay, so as I mentioned, we are going to be looking at the TCGA CLAD project. So you can see here on the top panel, we have listed out the name of the project. Here on the Dataset tab, we're showing all of the results that we've loaded in. All of these samples have been processed in a pairwise analysis. You see they're all paired. We have the patient ID in bold. That's the name that I'm using for the display. The data types for the copy number and sequence, quality score, and then factors or annotation. One of the great things about the TCGA data set is that there's extensive annotation that goes with every sample. And I've loaded just a few of these here. So these are what we refer to as factors. I'm just going to scroll over here so you can just see some examples of what factors may be. So in this particular project, I've loaded in the histological type, anatomical site of the tumor, tumor stage. I have some survival information, whether or not they have polyps, which was a risk factor, microsatellite instability, their gender, age at diagnosis. Really, you can load anything that you want in here. And the more information that you have, the more conclusions you can draw during your analysis. This is a project of approximately 220 samples, and this is basically all of the samples that had both this raw Affymetrix cell data and a corresponding MAF sequence variation file through the TCGA portal. Okay, so if we select all of the samples, which you can see they're all selected here, and click on View, we'll get the Results tab. And so here is the a summary of the overall results. So we're looking at the whole genome view. The top panel is showing the aggregate view of the copy number changes. We have red for loss, blue for gain. And then on the bottom panel, we have the individual samples listed. We have the display name here. And then for every sample, we have the gains, losses. We also have allelic imbalance events, loss of heterozygosity events. And then you'll notice these red dots. These are actually showing us any sequence variation that we have from the somatic mutation file. So the first thing that I always like to do when I'm looking at my results is to actually just take a quick browse through some of these samples to see if there's anything of interest. And I did notice a few interesting things that I just wanted to point out. So first, I noticed in this sample, there's on chromosome 12 here, it looks like there's some interesting calling going on. So what I can do is I can actually click on this blue link for the sample name and drill down to the probe level view for this sample. And that way we can actually look at the individual calls for the copy number and mutation for this sample. So you can see here if I click on the whole genome tab, and then let's make this big so we can see it. Okay, so we're looking at the rainbow view of the whole genome, and you can see we have the call for all of the different chromosomes, and chromosome 12 is the area that looks interesting. And one of the reasons this looked interesting to me is because the summary view had some hallmarks that indicated this might be a potential chromothriptic event. And in case you don't already know, chromothripsis has been recently described in a number of cancer papers, and it's basically chromosomal shattering. So the chromosome is broken apart into many, many pieces, and then it's reassembled sort of haphazardly so that you end up with large regions of non-consecutive loss, basically areas that don't get rejoined into this new chromosome. And one of the hallmarks of this feature is this aberrant gain-loss copy number calling, and also aberrant allele calling, and you can see the allele calls also look pretty strange here. So we can actually zoom in to chromosome 12 and look at this a little bit further. And I'm going to hide the table right here, so now we can just view chromosome 12. And what you can see is that we do in fact see this flip-flopping of copy number states from gain to loss throughout the chromosome. Looks like there's a good number of changes here, which typically defines chromothripsis. We see that in the lower states we do have loss of heterozygosity, which is good. So this might be an example of a chromothriptic event. And again, this is interesting just for general knowledge, but also because chromothripsis has been observed in a low frequency in many different tumor types, and it may have some implications with survival. So one other thing that I noticed when I was scrolling through this data is that there is a lot of variability in terms of the number of mutations that are called. So you can see this top panel doesn't appear to have very many mutations, but this one right here, this fourth sample, looks like it has a lot. And if we just scroll through, you can see that there are a number of samples that have a bunch of mutations. And this may be indicative of a hypermutable phenotype. So this is something I think we might want to look into in a little bit. But first, I want to also use the rest of the information that we have on the screen, and namely this aggregate view on the top panel. So after we've looked at some individual samples, it's nice to actually get an idea of what is happening overall. And so the top panel is showing us the aggregate summary for copy number changes. You can see we have, looks like there's some frequent gains in chromosomes 7 and 8. There's a frequent loss in 8p. 13 is frequently gained. Looks like 17p is frequently lost. 18 frequently lost. 22q has frequent gains, and X is gained in a good number of samples. We can also zoom in to look at some of these more focal gains and losses. So right here, if we actually want to just zoom in to this one, could be interesting. So you see there's a nice peak point for this loss. And if we hover over the genes, we can see that it's the F-hit gene. And that's actually a tumor suppressor. So let's flag this and put a pin in it and save it for further review because that could be interesting. So this is great for visualization, but what if you actually want to get a table of these results? To do that, we just have to click over to Aggregate. And now we're going to see a table view of our common regions of change. And this is showing us the maximal common regions of change in a frequency of greater than 35%, which is our default view. You see we have our region of change, the length inside a band, the type of change, and then the number of genes within it. So if we just scroll through this a little bit, we can see we have gains, we have losses. Let's just keep going. We have a lot of allelic imbalance that's being called, which could be interesting. But if I sort by first clicking on event and then control region, so I want to see if that that loss of F hit is coming up, because that could be interesting since it is a tumor suppressor. That was on chromosome 3, so we can see it's actually not showing up here. So two things that I'm noticing is, one, I'm very curious as to whether the F hit loss is in fact significant. And also I want to know if these allelic imbalance events that I'm seeing are significant as well, or if they are events that are co-occurring with copy number change, which would be less interesting. So first, to address the allelic imbalance, one thing that I can do is I can actually apply filters and filter out some of the data that might be less interesting for me. So by clicking on this funnel option here, it opens up the filters tab. So within Nexus 7.0, there's been a really great expansion of our filter options. Here we can filter based on copy number events, and this can be based on the literal calling of the event, or based on the size or number of probes, or whether or not it co-occurs with an allelic event. We can filter the allelic events, again, just based on what its actual literal name of it, the size or number of probes, or right here, we can remove all of the changes that are also copy number changes, which is what I'm going to select. This means that only the copy neutral allele changes will remain, and these are the ones that I would be more interested in for this analysis. We can also filter based on the sequence variation event. And finally, there's this area called region-based filters, and this includes removal of calls based on the sex chromosomes, or also based on your own customizable bed files, if there's a specific region that you want to either hide or focus in on. Okay, so now that I've applied those filters, it's going to apply that to my data and basically mask that from my view. So you'll see that the copy number there, you can see all of that purple that was associated with the copy number changes goes away. And now if we click back to the aggregate and sort by event, we can see that all of the allelic events that we observed, it did in fact correspond with copy number changes, so not interesting for us. Now, to find out whether or not this loss of F hit is significant, there's actually a really great tool to use called GISTIC. I'm sure if you've read the TCGA papers, you're familiar with GISTIC. This is a tool that's used to identify significantly enriched copy number changes within your data set. But instead of just doing this based on the frequency of the change or the number of samples changed, it also has weighted values based on the amplitude of the peak, meaning does it have a high copy gain versus a single copy gain, or a single copy loss versus a homozygous deletion. The focus of the peak, meaning is it a focal change that's really targeted to one gene, or is it a broad change covering an entire chromosome, as well as the overall frequency within your data set. And so to run GISTIC, you simply click on Tools and GISTIC, and then we can view those results. So here I'm showing you the GISTIC results. Let's make this big. Okay, so we have here the this is the way that GISTIC results are frequently reported within publications. The regions of change, extended region, type of change, and then the two metrics calculated by GISTIC are the G-score and the Q-bound. And I actually want to flip over to the genome view first. This is showing us on the top panel all of the copy number changes as an aggregate view, similar to what we saw in the previous results. But you can see here that in gray, it's showing the samples, or the areas of the genome that are significant. And we can view this based on both the copy number view or the Q-bound. And this Q-bound view is nice because this is the way that's commonly reported in publications. So if you want to change that. Okay, and we can also now look to see, I see there's this region on chromosome 3 that's showing up as significant, and I'd like to see if that is in fact the F hit loss. So one way to do that is to just sort by type of change, and then hit Control-click for region. And let's go down to the losses and chromosome 3, and we can just click on it. And it is, in fact, that F-hit loss. So even though it was it was found in an overall slightly lower percentage of samples, it is considered statistically significant. Okay, so now that we've sort of perused our copy number changes, I'd like to look at some of the results that we have with the mutation data. And the first thing that I'm really interested in is just observing this hypermutability phenotype and seeing if there's anything of interest here. So one way to evaluate this is to actually look at the number of mutations that are called with each sample. And to do that, I'm actually going to show you how to do this using factors. So one thing that I did when I downloaded the somatic mutation file, I also made a note of the number of mutations for each sample and saved that as an Excel spreadsheet, which I can now load as a factor. So I simply clicked on factors, load factors, and now I'm going to upload this text file that I made in Excel, which has the mutation count. Whenever we add factors, that data goes to the end of our spreadsheet here. So now you can see the information we have for all these different samples. So we have the total number of mutations, we have the breakdown of the different kinds of mutations, splice site, silent, nonsense, frame shift, missense. And here, this is the category I'm interested in, the total non-silent mutations. So within the TCGA papers, they've identified that there are a number of hypermutable samples across many different tumor types. And I'm going to add a factor called hypermutable here. Okay, and I'm going to rearrange my table. I can just click and drag to move it to a spot I want. So everything that's over 400 mutations is definitely safe to call hypermutable. And so what I'm going to do is I'm going to click on this option here, allow column selection. This allows me to actually select a specific cell instead of just the whole column. And everything that is over 400 non-silent mutations, right-click, set value, I'm going to call hypermutable. Okay, everything else I'm going to say is not hypermutable. Okay, and so now I've created a new factor here called hypermutable based on the total number of non-silent mutations in each sample. And so one thing that I'm interested in is I'm interested in seeing are there certain copy number or mutation changes that significantly that are common for this group. So one way to do that is to run a comparison. And so for any factor that you have within your data set tab, you can run a comparison. To do that, we simply click on Add Comparison, select the factor. So here it's going to be hypermutable. And again, this is showing me now all factors that we have added into the table. Our factor values are yes versus no. And this is going to guide me through creating my comparison. So you can see as I click on it, options that are options that are grayed out become available. So I'm setting my baseline to not hypermutable, and my experimental is yes hypermutable. And now we've created a comparison here. Now to view this comparison, we simply click on it and hit View. And you can see it's processing our data, and it's basically running a Fisher's exact test between these two sets to find statistically significant regions of change. Okay, and I'm going to click to the Genome tab first. This is sort of my favorite way to look at this. And I'm going to zoom in by selecting this scroll bar. Okay, and so what we can see here, the top panel is showing us the summary views of these two different groups. The bottom and how they are interacting with one another, and then the bottom is showing the aggregate results for each of these groups. So we have approximately a quarter of the samples are considered hypermutable, and you can see that they're really quiet in terms of copy number changes, but that there are some fairly frequent gains on chromosome 8. Versus the not hypermutable tumors, we can see that there's some gains, and you can see if I hover over it, I can get natural frequency. This loss on 8p is much higher, almost 50% of samples have this loss of AP. And also this gain of chromosome 13 is present in almost 70% of samples. So this is pretty interesting. We can also, if we click on, if we look up here, we can see all the areas that are considered statistically significant. And now if we go over to the regions, we can actually view this in a table format. So we have the names of those regions, the change that's occurring, the frequency in the yes group versus the no group, and then what's the difference between them. And if we sort based on events, we can see now these are all of the allelic imbalance events that do not co-occur with copy number changes. And if we just flip through here quickly, we have gains, losses, and we also have any sequence variants that are co-occurring here. So this looks like it could be a pretty interesting thing to look at. And one thing I'm always interested in determining, because we do have this with the TCGA data, is whether or not there's any impact on survival here for this. We know that that copy number changes often have a poorer prognosis. However, hypermutable samples probably do have a lot of mutations that could be deleterious. So we can actually look at a Kaplan-Meier plot of these results. Just right-click on the factor, okay, and let's look at overall survival. And it really doesn't look like there's much of a difference here today. So we can actually click on the significance, and it's really not statistically significant. So it could be this data set, or it could be that the mutations are affecting potentially deleterious regions. So we've looked at this data in terms of the overall mutational spectrum, but what about looking at some of the individual mutations to see if there's anything of interest? So what we can do here, I can remove this flag. We can actually change our view now to be the sequence variants. Okay, so now we're showing on the top panel an aggregate view of just the sequence results. And just like before, we can scroll through this data to see if there's anything of interest. So I'm going to look at this mutation over here. Chromosome 7 looks like it's in a pretty high frequency, and these are showing as a hotspot. There's a hotspot in BRAF. So this could be interesting because BRAF is known to be have altered expression within colorectal cancer. And it looks like there's also a really high peak over here on 12. Zoom in on that. And if we hover over it, it will tell us that this is KRAS, which is a known oncogene. Okay, so we can flag that for further review too. Okay, so I'm going to focus, I think, on the BRAF mutation because this could be really interesting. And like I said, there is known altered expression of BRAF within colorectal cancer. So one thing that we can do is we can actually ask ourselves within this data set, what are some of the copy number or mutation changes that co-occur with BRAF mutation? So there's a really great and fast tool that we can use to do this. If we click back to data set and look in the tools bar here, we can try the concordance function. So what this does is it brings up a query panel, and we can now type in a specific event and ask Nexus to identify regions that are significantly co-occurring with that change. Okay, I'm going to pick a region, and I'm going to focus on the sequence variant, and I just want to focus in on that BRAF hotspot, which I have right here in my notepad. Okay, and I can label it. So this is going to be the BRAF B600 hotspot mutation. Okay. Okay, and now what Nexus is doing is it's identifying those samples with the hotspot mutation and then identifying what are the copy number and mutation changes that significantly co-occur with one of these two groups. So again, let's actually just flip to the genome view first. And let's zoom in. Okay, so we can see that 26 samples have a mutation within that hotspot amino acid. 194 do not. You can see that that similar to the hypermutable phenotype, we do see that there is relatively fewer copy number changes in the mutant group versus the wild type group. Again, chromosome 8q seems to be gained in both. We can see that the gain on chromosome 13 is much higher, as is X, and these areas of loss are much greater in the wild type areas. And if we look at our significant panel, we can see that which changes are statistically significant between the two groups. Now, if we actually click back to the regions, you can see that there are a number of regions that are significant. I'm actually going to narrow this down to just the peak of the region. And so now we can look at this table a little bit more closely. So here we have a column for display, and this is going to, and label, and I'm going to come back to these in just a minute. We have the region of change, the location, the type of change, length of the region, frequency and queried event. So this would be the frequency of that change within the BRAF hotspot mutants versus the frequency of the BRAF hotspot wild type samples. The overall difference, a p-value that can be calculated based on a probe level, overall p-value, a q-bound for a multiple testing correction, and then percent of CNV overlap. Now just to jump back over here quickly, one of the great things about this feature is that we can actually visualize these changes using a circular plot, which right now is empty because I haven't selected anything. But this is a great way to visualize a lot of data in a very small amount of space. So let's create a circular plot of some of these significant regions with the peak areas. So one thing that you might have noticed is that we actually don't have any information right now about the genes that are in these regions, and that's probably where the interest is going to be. So I'm going to click Modify View and select gene symbols and microRNAs for each of these regions. There's also a number of custom tracks available, and I'm going to scroll down and select this Sanger Cancer Gene Census, and I'm going to click IDs because I want to know if there's any known cancer genes within this data. Okay, and I'm going to click and drag each of these columns over so that I can read them easily. Okay, and now I'm going to select areas of interest. So first I'm going to sort based on the event, and I want to select all of the mutations that co-occur with BRAF hotspot mutations, because I think that's pretty interesting. I'm also interested in any microRNAs that co-occur and any cancer genes that co-occur. Okay, and next I want to actually sort based on this differential. So I'm really interested in any changes that are occurring at a very high differential frequency, greater than 50%. Okay, and you see this has a negative value. This means that it's higher in the not queried group or the BRAF hotspot wild type group. And I'd also like to include anything that's significant only in that BRAF, any copy number changes that are significant only in the BRAF hotspot mutant group, which includes this RBF XO1. Okay, so if I click on this display button here, it's actually going to show me all the ones that I've selected. The label is showing us the genes that we've picked, and now if I click on circular plot, we can view these results. We can actually zoom in here and move it around. Okay, so we see here we have our query, which we have labeled the BRAF V600 hotspot mutation, which is right here. We have all of the statistically significant co-occurring events. We see, you can see here we have black is indicating mutation changes. Blue is indicating gain, so red is loss. We have black solid bars to indicate things that are significantly concordant with the with our query, and anti-concordant, meaning that they're specifically enriched in the group that does not have that change. Okay, now one thing that I've noticed over here, it's showing me now a key for all of the genes within this region, and for this one I specifically wanted to really just refer to the microRNA. So what I can do is I can click back to the region and edit right in here. That just says a microRNA. And there's also this gene right here, which does, or this region right here, which doesn't have a gene. A known gene. So I actually want to make a label that says no known genes right there, just so that way I know that when I'm showing this to other people that they don't think there's something missing. So I can make edits into this label area, and when I click back, now you can see that my edits have been applied. And so now I have this really great figure to identify statistically significant regions which are co-occurring with our hotspot mutation. Now, of course, this is all well and good, but I want to also wonder, is this functionally relevant? And one way to do that is to actually look for enrichment. So what I can do within the Regions tab is I can actually highlight all of these things that I have selected here to visualize and click on this Enrichment button. Now what this is doing is it's taking the gene list for this set and running enrichment testing to see if there's anything that's statistically significant. Make this just a little bit bigger so we can see it. Okay, so in terms of biological processes, there is one that's significant both based on the p-value but also following multiple testing correction, and this is kinase signaling pathway. If we click on the molecular function, we have a few that are of interest, and one of them is its activin receptor activity. We also can look at the KEGG and biocarta pathways if we're interested in looking at those. There are actually a number of different enrichment options that you can choose to customize to your results. So it looks like this actually could be of interest. And of course, I can always export these results as a table if I want to share this with others. Okay, so we've identified that there is something going on with this BRAF hotspot. There are significant changes that co-occur with it. It looks like it also has some interesting phenotypes in terms of the copy number changes that go along with it. What if we want to see if there's any survival associated with this as well? Well, one way to do that is by adding it as a factor. You remember when I made a factor for the hypermutable phenotype, I could right-click it and identify, create a Kaplan-Meier plot. So we could go through and find all of the BRAF mutations one by hand, but that sounds like a pain. So what we can actually do is use this query function. So when I select query, now I have a query panel here, and I can search for any kind of change I'm interested in. Here I'm going to do it based on region because I want to identify that hotspot. Click Run, and now you can see very quickly, excuse me, I have all of the samples within my data set and then my query event, which was this region, and any changes that are occurring. And I'm just going to scroll through here. I want to select all of the point mutations that are within there, and also these two right here. Okay, so now I've identified all of the point mutations. Now I can actually hit Apply Selection and close this, and what you'll see over here is my selected samples has changed from all of them to just 26. So now it's just selecting those 26 that have the change. I'm going to add a factor here and call this BRAF B600. Okay, so I'm adding a factor that's for the hotspot. Okay, and now slide this over to a place where we can see it. Okay, so I can sort based on the samples that we have and add as a factor. I can actually label, mark all of these ones with a yes. So these are all the ones that have that hotspot mutation. And these are all of the samples that do not. Okay. And now I'm going to actually select all of the samples. Okay, and I'm going to see if there's any survival associated, or what the survival is that's associated with this BRAF hotspot mutation. So I just right-click and select the Kaplan-Meier plot, and you can see this actually looks a bit more compelling than the last Kaplan-Meier curve. If we hover over it, we can see the red is for the BRAF mutant, and the N is for the ones that were wild type at the hotspot. And it looks like there's a pretty significant difference here. We actually click over to significance. It's not quite statistically significant, but it's much closer than the last one, and it's definitely something that we could flag for further review. Now, if you'll recall, when we actually were looking at the copy number changes for this group, it also looks relatively flat, like the hypermutable phenotype. We can actually run a factor enrichment here, which I'm just right-clicking and now clicking factor enrichment. And this is going to actually allow us to determine if the factor values are overrepresented with another factor. Or in other words, are these two factors very similar to one another? So here we have the factor here is listed, so this is showing all the other factors, and specifically hypermutable. And we have the factor value, yes or no, the number of the factor in the group or in the selected samples, the percentages, and lastly is the p-value. And so what you can see is that this p-value is listed as 0. It's actually not 0. It's very significant, so it's almost close to 0, but it's highly significant. And so this is indicating that the hypermutable factor is enriched among the BRAF mutant group as well. And this is not necessarily surprising. This may indicate, though, that most BRAF hotspot mutations are also hypermutable. And this is just one way for us to look at that. Okay, so I think with that, we'll wrap up, and I'll take any questions. Just in summary, I've shown you today the integration of mutation and copy number information using the TCGA data set, specifically with the colon adenocarcinoma paired project. I showed you the aggregate and GISTIC functions for identifying both large regions of common change as well as specific enriched regions of focal events, which included that F-hit deletion. I showed you some of the enhanced filtering options, which included removal of the allelic imbalance, loss of heterozygosity events that co-occurred with copy number changes. I showed you some of the comparisons that we can run using loaded factors, which included hypermutability in this case. I showed you the new concordant function with the BRAF hotspot mutation, survival analysis with both the hypermutability and BRAF, and then finally factor enrichment. And so I think with that, I will take any questions. Thanks.